Hello, my name is Paola Picotti and I'm a professor of systems biology at ETH Zurich. I will now let Valentina Cappelletti, a scientist in my group, tell you about our research. Valentina was a major player in the project we would like to tell you about.
Hello, my name is Valentina Cappelletti and I'm a structural biologist. I study how the 3D shape of proteins regulates their function, and I do this for thousands of proteins at the same time. Our research breaks the wall of measuring changes in the shape of thousands of proteins at the same time and with their physiological context. Our laboratory has developed a new technique to do this. We can now tell which proteins have acquired a changed shape either during normal functioning or in disease. Proteins are the building blocks of life. They form our cells and organs and serve many important roles, for example, as hormones or antibodies. They come in different shapes and form complex and fascinating structures like this one. This model shows a protein which protects bacteria against heat, and it's of course a magnified model. A real protein is much smaller. We all know that the shape of an object is important for its function. For example, the circular shape of wheels ensures that a car can move smoothly. Similarly, proteins need to have a specific shape to function properly. This is the so-called structure-to-function paradigm. The structure of a protein is, for example, responsible for the interaction with a partner molecule, which can bind the protein and activate it. Sometimes proteins fail to acquire the right shape, and this structural change may cause the protein not to function anymore. For example, not being able to host its partner anymore. Incorrectly shaped proteins have been linked to a number of human diseases like Parkinson and Alzheimer, showing the importance of studying protein structural changes. Let me put this in the context of recent scientific breakthroughs. In June 2000, the Human Genome Project made it possible to read the whole collection of human genes. 14 years later, we could read the Human Proteome too, the whole collection of human proteins. While this is a powerful approach, it has a major flaw. It is blind to the structure of proteins, but only tells us how much of each protein is present. And this is where our technique comes in. We can now measure structural changes of the whole collection of proteins in an organism. So for the first time, we have a type of molecular data that can tell which proteins have changed shape between healthy and diseased states, thus hinting at what has gone wrong in the disease condition.
Our analysis looks at biological systems with an entirely new type of lens. We have recently used this lens to look at alterations in the bodies of Parkinson's disease patients. But it can be applied to any other disease or condition, ranging from cancer to antibiotic resistance. So we think the implications are many. We now would like to integrate artificial intelligence with our technique to generate new valuable knowledge in the field of personalized medicine.